All right. Well, the original topic I realize of this session is supposed to be ninja skills. And so I'm going to show you some ninja skills, but it's not going to be my standard ninja skills presentation where I usually start out with compensation. Instead, I'd like to introduce you guys to a workflow using some of the plugins that we have available in FlowJo version 10 to discover what populations change between a set of samples that have been stimulated with a TLR agonist. And so I call this a discovery workflow. And I'm going to use the process of downsampling, concatenating, that's merging samples together, dimensionality reduction using an algorithm called T-SNE, and clustering using an algorithm approach called FlowSum using FlowJo version 10. So this is stuff that you can do in FlowJo right now. Just a warning that the FlowSum algorithm does take some setup, and I'll briefly go over the setup of the R statistical computing environment and then point you to some resources online on our documentation page where it explains in more detail how to set up FlowJo to interact with R and utilize some of these algorithms that the community is developing in that R statistical computing environment. So to start, I'm going to go through these slides, and what I'd like to do is give you an idea of the workflow, and then I'm going to demo it live in the workspace and explain things as I go through and give you an opportunity to ask questions. And so again, if you have any questions, one thing you can do is raise your hand, so it'll give me an auditory beep to tell me that you're typing something. But we'll have an option, and I'll turn on all your mics here at the end so that we can have a discussion about this. So in this example, I've got two patients, and I'm developing an assay. I want to look at the response of peripheral blood mononuclear cells that have been frozen and stored and brought into my lab. And in particular, the HLA-DR positive antigen presenting cell compartment within the PBMC to a stimulation with a TLR agonist cocktail. So what happens, what populations are induced when I stimulate with TLR? Each of these patient samples was thawed out of the liquid nitrogen and rested overnight to kind of let the cells settle and recover. Anything that's going to die dies off. And then you know that you're left with healthy cells. If you're doing a stimulation, it's good to rest, and you'll get a better response after even just a couple hours. But I usually rest overnight and then process the next morning. So then in the morning, I process in parallel with or without stimulation for an 8-hour stim in this case. It's always good to do a time course depending on the cytokines you're looking at, but in this case, I'm using an 8-hour stim. And then I've stained with a 13-color panel using a standard intracellular cytokine staining protocol. So I'm not trying to get down to the nucleus. I'm just looking at cytokines within the cytoplasm. And so the perm buffer that I use is not very harsh, but it's this typical cytofix cytoperm protocol from BD. And then I collect the data. In this case, it's on an LSR2, an 18-color instrument. And I've actually collected all 18 parameters, even though I've only using 13 channels or staining for 13 colors, because you can always do spectral compensation on any instrument when you have more parameters than you do colors. And so I've been looking at that to improve spillover spreading using spectral instead of standard compensation. But that's off topic, I'm sorry. My goal here is to identify what phenotypic populations are induced upon the stimulation with TLR without really knowing what I'm looking for. And I'm going to show you how I would normally approach a gating tree as well, but this approach would be if I'm trying to find out what's going on and I don't know specifically what the phenotypes are that I'm looking for. So I'm not going to use a basic gating tree for specific phenotypes to analyze this data alone. Instead, I'm going to use a clustering algorithm, and that clustering algorithm is FlowSum, as I mentioned. I'll go through that in a little more detail in a moment. But that algorithm will create these populations or clusters of events, cells that have similar phenotypes. And then I'm going to determine if those clusters or populations are differentially expressed between the no-stim and the TLR stimulated experimental conditions and determine the phenotypes of those clusters. So for the clustering algorithm, I'm going to use this plugin called FlowSum. It's available off of the FlowJo Exchange website, which is the very first link. If you go to the Workspace tab, and then look at the plugins menu within the population band here in the workspace tab in the FlowJo version 10 ribbon, you'll see a drop-down menu called plugins. And standardly, we release 5 plugins with the release that's downloaded with FlowJo. But FlowSum is a new plugin that we have released just in the past month or two, and that is available for download on the FlowJo Exchange. And I'll show you that when I go into the program. But when you initiate this plugin, FlowSum, you'll be presented with a GUI, and you can specify the number of cluster populations, or meta clusters in this case. Right here I've specified the number 20, and the parameters that you're going to use to cluster within the file if you want to limit the parameters. And of course, in this case, I'm only using compensated parameters, and I'm not using the ones that I've already gated on, my dump gate and my viability gate. So I'm using 11 out of the 13 parameters in this case. And when I select the parameters, tell how many clusters I want, and then click OK, FlowJo sends this data file over to R, and R does a calculation. It returns a bunch of information about the populations. And the end result of the FlowSum calculation is the cluster populations here, FlowSum Pop 0 to FlowSum Pop 19. So I've got 20 clusters, as well as a visualization of a minimal spanning tree of the meta clusters and subclusters. And then we have these kind of cool star charts, the wheel charts that show the relative frequency of the individual markers that were used to calculate the tree and the cluster populations. And so there's a reference here, Van Gassen et al. From 2015 is the actual paper if you want to understand more detail about the math behind this clustering algorithm and the visualizations associated with it. But once you create the clusters, if you drag the FlowSum node that is created over to your layout editor, it will render this tree with the star diagrams and a color-coded map for what clusters relate to the populations over here in the workspace. The other tool that I'm going to use, which is not as much for analysis as it is for visualization, is the dimensionality reduction approach of T-distributed stochastic neighbor embedding, T-SNE. And this is an algorithm that we perform to dimensionally reduce high parameter data and allow visualization of this complex multi-dimensional type of data set in a 2D plot. And the key here is that when you dimensionally reduce using T-SNE, the events are redistributed along two new parameters, two new axes that are created, derived parameters, T-SNE X versus Y. And it creates this continent-like structure, but the relationship of the events to each other in the n-dimensional or the multi-dimensional space is maintained in this two-dimensional projection of the data. And so here I have taken a 15-parameter immunophenotyping file, and I've dimensionally reduced it to create these X and Y parameters. And then I manually gated known populations and overlaid them in the layout editor of FlowJo to create this diagram to show essentially that the phenotypic populations separate with the algorithm. In green, all the green cells are CD4 positive T cells. However, this dark green node that clusters together as a set of events is actually the Treg population, the CD127 negative 25 positive population within the CD4s. Similarly, you've got the CD8s in brown, and then you've got a node over here that is CD127 27 positive population, so some maturation subset. Then you've got natural killer cells in red and B cells in blue, and they parse out or move to and occupy distinct regions of this continent structure, the T-SNE data space, I like to call it, because of their similarity in phenotype in the complex multidimensional phenotype that we're interrogating. So one of the things about T-SNE is the key to comparing different samples within a dimensionally reduced data space is that you have to run the T-SNE algorithm on all the data together, not individual data files, because it's a non-deterministic algorithm. It takes one event and it seeds the algorithm, and then that event talks to or picks 20 or 30 or 40 events surrounding that event. And each of these events pushes and pulls on each other until this redistribution occurs. And you cannot control that seeding and the picking of the other events that each event talks to in the algorithm. And so you get a different representation every time you run it. Maybe it'll look similar, but the orientation will be different. And if you run on completely different data from one sample to another, you really don't get the same comparable dimensionally reduced data space. And so we're going to first concatenate or merge the samples together into a new FCS file and then run the T-SNE dimensionality reduction step so that we can pull apart the individual component files and compare them within this common dimensionally reduced data space. And so I know this is a long slide, you don't have to read the whole thing, but this is the workflow for this. I'm gonna first bring in my data. I'm going to compensate it if I haven't done so. I'll start with compensated data today. Then I'm going to clean up gate which means I'm going to get rid of the dead cells. I'm going to get rid of the doublet cells, get rid of any particulate matter that's not a full, complete cell, so pieces of membrane and stuff, debris. And just wanted to point out here at this cleanup gate step, we do have a very cool set of plugins, Flow AI and Flow Clean. These are algorithms that actually will pull out the bad data from a file. So it'll get rid of clogs in your samples and remove those events that were not consistent with the along the entire collection. And these are automated approaches to clean up your samples if you have clogging issues that have been published and are available as plugins at the FlowJo Exchange as well. So after I clean up the sample, I then am going to take my target population. In this case, it's going to be DR positive, HLA-DR positive antigen presenting cells that are not T cells, not NK cells, and not, what else do I dump? And not B cells. And then I'm going to take those target populations. When I've downsampled them, I'm downsampling to limit the number of events that go into the T-SNE algorithm because it's a very memory intensive process. So I'm going to concatenate the target populations together into a new file. Then I'm going to use the T-SNE dimensionality reduction algorithm to create this dimensionally reduced data space. And I can visualize this T-SNE space, I got that duplicated unfortunately, with a third parameter color mapping. And I'm going to show you how to make a query gate overlay in the layout editor so you can see what phenotypes, what the complex multicolor or multi-parameter phenotype of your population in the query gate is, and you can move that query gate around and explore this dimensionally reduced data space. I'm then going to gate on the populations, gate populations based on their experimental conditions, so gate stim versus no stim, and then use that clustering algorithm to create populations. I don't know what they are quite yet or whether they go up or down, but using the experimental condition gates, I can drop those onto the cluster populations and then classify whether those populations are enriched in the stimulated or unstimulated condition. And of course, then go and look at those populations and phenotype them either in an all histograms overlay in the layout editor, or what I like to do is export the populations and add all of the medians of the compensated parameters. So I get all of the MFIs for every channel for every population, and I'll batch that in the table editor to get all the medians of all parameters. And so let me show you in a visual way with slides what I mean by all of those steps. So first I'm the cleanup gate. In this case, I got the 4 samples. I usually do a time gate, so in case there's a clog, I can move upstream or downstream of that, but I've been going towards replacing this time gate with an automated Flow AI or Flow Clean process that would get rid of any clogs and then stitch back together the good data, so you preserve a little more data in that mechanism. But the time gate is a standard way to allow you to get rid of bad events if you need to. And then I'm going to make a singlets gate, so forward scatter area versus height. Cells that are on the diagonal are single cells, and then things that are off the diagonal are doublets. And so I'm going to keep everything on the diagonal and get rid of these events that pass by the detector 2 at a time. Then I'm going to do a size or a whole cell gate, and I'm essentially using this gate to get rid of the debris that's in the very small forward side scatter region down in the corner. And then I'm going to do a viability gate. So it's always good to have a viability die no matter what samples you're running, but especially if you're going to freeze thaw samples or digest tissues, then you're going to have some dead stuff. And so getting rid of that dead debris, which happens to usually can be very sticky and lead to non-specific binding of antibodies, cleans up the data and gives you the functional cells that you know are actually going to be intact and give you good readouts. So after the viability die, I'm left with live cells, but I still have a whole bunch of cells that are not antigen presenters that I'm not interested in. And in this case, I'm going to dump C3, 56, 19, and 20. So this is a dump gate in a single color with antibodies to all 3 antigens, or all 4 antigen markers here. So I get rid of multiple lineages in a single color, leaving my other channels open to look at the phenotypes and functional responses. Final cleanup gate is to isolate just the DR positive events. So I'm going to gate only on DR positive and get rid of everything else. So this is my antigen presenting population that I'm calling DR positive here, highlighted in the workspace samples pane as a DR positive gate. So that gives me clean populations. And now I'm going to look at their phenotype downstream. But before I do that, I want to limit the number of events that I'm going to throw at this T-SNE algorithm because it's a very memory expensive algorithm. And if I'm really doing discovery and I have time to wait half an hour for the T-SNE run to complete, maybe I would downsample to 20,000 events per sample. And then that normalizes the number of events contributed from each file, which is important so we know what we're starting with to determine whether populations are changing. But it also is reducing the amount of memory it's going to take on my computer and the time it's going to take to complete. In this case, I'll probably just downsample live to 5,000 or maybe even 3,000 events so that this T-SNE step takes just a couple minutes. But that is the bottleneck there in terms of memory and calculation speed, is that you can, depending on how good your computer is, you can only throw so many events at the T-SNE calculation at a time without running out of memory. So after I downsample, this creates the, downsample is a plugin. It runs natively without no additional setup, and it brings up this window. You tell it how many events you want to include, and then give it a population name, and that becomes a population down here that has the specified number of events. So I can do that for each DR positive cleaned up population, and now I'm left with 20,000 events in each drpos.pop downsampled subpopulation there. Once I've downsampled, now I want to actually concatenate or merge the populations together. So I'm going to highlight all of the downsample populations here, all 4, and merge them together using a platform called Export and Concatenate. It's located under the File tab in the Workspace ribbon. And I select Export Concatenate Populations because I want to only get the events within the downsample pops. And that will open up this interface, this platform, where you have 2 tabs, Export or Concatenate. If you have Export selected, that would generate new sample files for each selected population. But if you select Concatenate, then that will merge the selected populations together. And in doing this, I'm actually going to create a numerical value that specifies the order and or the experimental condition. So I'll give the no stims a number of 1 and a stim the number of 2. And what I'm going to do is I'm going to actually create an additional new parameter that separates the stim samples from the no stim samples. When I concatenate that file, it'll write that file to disk, and then I can check the existing workspace button here in the export message and close this interface, and that will load that concatenated file back into my workspace. And I am then ready to do my T-SNE and my clustering algorithms. So once I have that concatenated file, I'm going to then use the T-SNE plugin, also available under the plugins menu, under the workspace tab, and runs without any additional setup. T-SNE will derive 2 new parameters and redistribute events within this 2-dimensional space that I call the dimensionally reduced data space. When you select T-SNE, you'll get a interface where it prompts you to choose the parameters you want to use for dimensionality reduction. And then you have a couple options in here for iterations, perplexity, ETA, and theta. I would only recommend playing around and changing the iterations and perplexity fields unless you go and learn the math and understand exactly what ETA and theta are doing. Iterations is the number of cycles the algorithm goes through. You can limit the number of iterations and still get a very nice plot, or you can let it go for longer and you'll get better separation, or even if you let it go too long, it kind of fragments into a bunch of islands. But there's a sweet spot for every data set, and it's different for every data set depending on the data. So we give a default of 1,000 iterations. I usually try any new data set running at like 600 or 800, and that is sufficient to give me good separation. The perplexity is the number of events that each event is talking to, the number of neighbors that it's talking to as it reorients and communicates between them. So as the algorithm goes through, if I have more parameters, I'll up the perplexity from 20 to 40 or to 50. But generally, you're going to get good separation even if you just use the defaults that are presented to you without changing any of these options. The key is to only use compensated parameters, right? And again, I'm removing the viability and the dump gate parameters because I've already gated on those, and I don't want those to influence the separation of events in this 2D space. So when I get done with this calculation, it takes a couple minutes, or even on my computer with 80,000 events in this case, it would take about 20 minutes. I end up with these parameters, and I can distribute the data across the T-SNE data space. Now, within this graph window that we have, there is a checkbox here for color map access down below the x-axis label. Turning that color axis on, then you can select a new parameter, a third parameter, to heat map or color map across the two parameters that you're viewing here in the graph window. And so what I've shown here is me turning on color mapping, and then going through and visualizing each of the measured parameters for each marker across the T-SNE data space. And you can see the distinct regions of expression are labeled for each marker as the red, or red is higher expression and green or blue is lower expression. You can see that the markers occupy distinct regions, but that there's this node over here that actually has a combinatorial expression with high CD1C, high TNF, high L1 beta. And so by using the color map, you can kind of see where everything is going, or where the antigens are expressed within this T-SNE continent structure, and get an idea of maybe where the co-expression is. Now, I've also gated on the TLR stimulated versus no stimulation, and I've made graphs of the T-SNE plot here in pseudo color. So you can see the TLR stimulated events occupy one region, and then most of the no stimulation events are occupying a different region. There is some overlap, but what seems to be occurring is that when you stimulate the cells, they change from whatever this phenotype is under no stim over to a different region, because their phenotype, they're upregulating all of the cytokines and changing their phenotype, so they move or redistribute to a different area of the plot. And that is indicating a large shift in phenotypic information within this panel here. So the total TLR stimulated is one region, and no stimulation is another region. So I know something important or big is going on here to move these cells over to a different region. What I don't know is what populations these are that we start with, and what do we end with, and that's what I'm going to try and figure out. And so when I'm in this data space in T-SNE, I can make a query gate, which I just circle a region that I'm interested in, and draw a gate. So it gives me the frequency of events within that gate. But then what I'll do is take a look in my layout editor and drag on the root file. So I have all events in red here, and then the query gate, I drag and drop it on top of all events to create an overlay plot where blue is the target population within my query gate. If you right click on this plot and an overlay, at the bottom there is multi-graph options. One of those multi-graph options is a histogram, multi-histograms. And here I've just displayed the 11 phenotypic parameters in a histogram plot profile for each of the parameters, and am comparing and able to visually call the expression pattern of the area that is within the query gate. So this is IL-6 positive, DR high, CD1C low or intermediate, TNF rocket high, 83 positive, IL-1 beta high, IL-12 intermediate maybe. It's negative for 123 and IL-10. And CD14. So, I have a phenotypic way to explore this. Now, look, what's cool is that if I have this graph window, I can grab this query gate and move it around to any region, and the overlay, along with all the associated histogram plots, update. So I can just explore this data space, this dimensionally reduced data space, and find out what this node is, and what that node is, and what that node is, using a query gate with an overlaid histogram. Now, in order to determine whether my query gate goes up or down in any region that I place it. So if I move the query gate around, I can move it here, and I want to know, well, is this area enriched during the stim or the no stim condition? And so to do that, I'm actually going to go back to my concatenated file and display along the x-axis my sample ID parameter, which is created during the concatenation step, and it separates all the individual files that were originally concatenated together. And so the two on the left are the no stim samples from the two patients, and the two on the right are the TLR stimulated. And so I can gate on stim versus no stim. And then I have these gates available, and I can paste them or drag and drop onto the query gate. And here, in this case, the query gate, wherever it be localized, is showing me that the no stim samples contribute almost no cells to this query gate phenotype. But the TLR stimulated samples are 99.9% of that query. So obviously, whatever is in that query gate, wherever it is right now, is enriched or induced by the TLR stimulated condition. And so this gives me that answers that question of whether or not this population in the query gate is increased or decreased between the experimental conditions. So instead of making a whole bunch of query gates and chopping manually that entire data space up, I can then go and use a clustering algorithm to redistribute the population, or to create populations that I will then determine whether they go up or down and what the phenotype is. And so, as I showed before, plugins menu, FlowSum. This FlowSum plugin is an R-based plugin, so it does require setup. But I'm going to tell the algorithm the number of clusters that I want to produce, the number of populations, and then click OK. And it sends the data R, returns all these cluster populations. And then once it's done, I can drag the FlowSum node that's created over to my layout editor and see the minimal or the spanning tree that's created along with the phenotypes within the meta clusters and the smaller clusters within them. At that point, I'm going to take those experimental condition gates, that's the stim versus no stim, that I know I started with 50% of each going into the total concatenated file, and I paste them onto all of the FlowSum populations. Then I'm going to go through and I'm going to classify, did these populations go up or down, and look at each one. So FlowSum pop 0, is it enriched under the TLR stim? Yes, then it goes into the overlay that's increased in TLR stim. The next population, if it's up in that stim, then I drop it on. And I can build this overlay. Again, right click, down at the bottom, we have ability to show multi-graph histograms. And I'm just looking at the 11 relevant parameters that are the phenotypic and functional information about these populations. You can see the vastly different phenotypes associated with these different cluster populations that were created by FlowSum. But every one of these populations is enriched to a great degree upon TLR stimulation at the expense of something else. So last step in my workflow is to find out exactly what the medians are, the phenotypes of those populations in more detail, not just visual assessment, but I want to compare, like, what is the expression of each of these populations that I've defined in every phenotypic parameter that I've got available to me. And so what I've found to be the easiest thing to do is to actually select all the populations and export them. So remember, exporting generates new files for each of the populations, as opposed to concatenating, which merges those populations together into a single sample. So in this case, I make 20 new data files for the 20 populations, and then I can load them right back into my existing workspace. And on those new files, I drop the medians of all compensated parameters. So this is from, I guess I don't have the right tab up here, but the workspace tab is there's a statistics band, and you can select all compensated parameters, and that gives me the MFIs of this population for every population. Put those into a group, and then I can go ahead and use my table editor to visualize and heat map across all the variation of each marker across all the FlowSum populations, and essentially go through and say, oh, this is positive or negative or intermediate expressing for each of the markers based on the range that I see for all of the populations. And so that's going to get you the more specific phenotypes of what is this positive or negative or intermediate for every single marker. And so these algorithms, FlowSum, the cleanup data algorithms like FlowAI and FlowClean, these are all available on the FlowJo Exchange GitHub site. If you click within the workspace under the plugins menu for the FlowJo Exchange, it'll bring you to this site, and this is where we release new plugins and we provide updates, and there's also developer documentation if you want to create your own plugin with some external environment to bring data in and out of FlowJo and perform calculations that are extra in addition to the normal FlowJo package. We have searchable documentation at docs@flojo.com. And if you go to our docs site and search for plugins, there's the first page is a installing plugins page where it describes how to set up the plugins, where to download R, and how to initiate the unpackaging or installation scripts for the plugins that depend on R. So I'm going to jump out of this workspace, and I'm going to go through that. Let's see if we have any questions. We got a few people. Okay, so here I'm looking at a workspace. I've already brought in my 4 samples, right? And these samples are previously compensated. And I've gated down to my DR positive population. You can see that if I want to do this manually, I'm going to be creating some more gates, including all the subpopulations that I can think of for the different antigen presenting cells. And then I might actually go and do Boolean combination gates downstream of all of these individual markers to see what the combinatorial expression is. So the gating trees can get really long when you're working with lots and lots of samples. And so I'm going to start by taking this DR positive population. In some of the samples, I've got 130,000 cells. In others, I've only got 21,000. And so I'm going to reduce the number of events, creating a downsample population for each of these DR positive nodes. And so the mechanism here is to select the population you want to downsample, go to your workspace tab, plugins tool, and drop-down menu, and downsample. It says how many events do you want. I'm going to go ahead and do 5,000 today, and I'm going to call this DR pos as a gate. So it downsamples, and then at this point I can actually distribute the downsample node to other populations. So I can drag it onto the next node, or I could drag it onto the gating tree so it would apply across all the samples within the gating tree automatically when those samples go into that group. And doing it like that, I've just got these populations. Now I've got that 5,000 events from the DR positive population uniformly downsampled into new pops. So the next step, I want to select all of these populations. And yes, I can hold down a modifier key like Command here on my Mac and multi-select each of those populations like that. But if you have 20 populations or 100, whatever number of samples you're working with, it's almost much easier to right-click and then select the equivalent nodes, which selects all of the populations that have an equivalent name and level in the gating hierarchy. That is available as a button up here in the Edit tab, Select Equivalent Nodes, but I always do a select the first population, right-click, select equivalent nodes, and now I've got all the populations highlighted that I need to merge together. So now that those populations are highlighted, I'm going to go to my Export Concatenate Populations tool, which is under the File tab, Document Band. Here it is. When I initiate that, it opens up my Export tab. So I don't want 4 new data files. I want to concatenate these together. And I actually realized I missed a little bit of a step that I want to show you before the concatenation step. So I'm going to close that dialog for the moment. What I want to do here is I actually want to be able to pull apart my samples between the stem and the no stem samples. And so I'm going to collapse this gating tree, go up here, and initiate a new keyword. A keyword is a metadata label describing the samples, and I'm going to call this keyword stem condition, right? When I create that new keyword, it creates a column that I can fill in or label information about this. So I'll give numerical values of 1 for the no stem and 2 for the stem, so I can actually separate them out into stimulated versus unstimulated populations. And it'll make more sense when I show you the concatenation. And I'll go ahead and grab the downsample populations here. Again, I'm right-clicking and selecting the equivalent nodes. Then I'm going to go to File, Export Concatenate the populations, and instead of exporting, I'm going to merge them together with concatenate. It's going to write on my desktop. That's the destination. I have some advanced naming options. I'm going to say DC stem. Just give it a unique identifier here when I write it to disk. So, cap locks around. And now, last thing I do before initiating concatenation is I'm going to choose my keyword that I've just created to create a new parameter based on that keyword. So I'm going to select the Choose button here, under additional parameters, scroll down to my asterisk keyword, which is right here, stim condition, and click OK. So now I'll get a new parameter in addition to sample ID, which separates all of the individual files. I'll get a stim condition parameter, which will separate into two groups of sample events, which is stim versus no stim. I'm going to concatenate that file. I'm going to load it back up into my existing workspace by checking the box and closing the dialog. And now I have 5 samples instead of 4. If I scroll down, here's my new sample, the concat1 DC stim that I just created. So double clicking on that sample, you can see that I've actually got a sample ID parameter that separates based on, let's see if I go to something that's functional, based on the order of the samples in the workspace when I concatenated. So there's 2 no stims and 2 stim condition samples. But then I've also got a stim condition new parameter that separates into 2 populations. It merges the 1s and the 2s, whatever samples were labeled with a 1 and a 2. So I can say that these are the stem cells and those are the unstem cells. And so whether I gate for experimental condition using this view or that view doesn't matter here. But if you have 10 or 20 or 30 patients under each condition, or even multiple conditions that you're comparing, it really helps to add that condition keyword and create a parameter that merges the multiple files under the different conditions together into single populations, and it makes it easier to gate on stim or no stim. So this is no stim, and I'm going to call this TLR stim. Right, so that's my gating for experimental condition. Now I'm going to close this, and I'm going to go ahead and initiate my T-SNE my T-SNE dimensionality reduction algorithm on the concatenated file. So I'm going to go to plugins, use T-SNE, and here it's prompting me to choose the selected parameters. So I'm going to choose, again, all compensated parameters. That's what's selected by default, but I'm deselecting the viability and the dump gate channels that I've already gated away. Cells that are positive in both of those. The iterations doesn't really matter. I'll do 800 again, and I'll do a perplexity of 40. I'm using fewer events here, only 20,000 instead of 80,000, the example I was showing you. But I'm going to go ahead and choose the selected parameters, and now it's going to show me that the T-SNE algorithm is calculating, and I'll be able to there we go. And I'll be able to monitor. At some point, this is going to stop calculating, and it will show me the 2 new derived parameters. And then at that point, those will become available for me to select within, was on stim condition, will become available for me to select here within the drop-down selection menus and the X and Y axis labels of the graph window. So while that's calculating, I'm going to go ahead and open my layout editor, and I'm going to kind of pre-prepare some stuff showing you how this works. I'm creating a new layout here, running out of screen space. New layout, and I'm going to call this T-SNE. Bringing in all 3 of my gated versus ungated populations here. And I'm really just killing time for the moment until that T-SNE completes. But I'm going to actually start with the gating, duplicating these populations. As soon as that T-SNE is done, I'll be able to change to the X and Y parameters and view the distribution of events. And then I'm going to make an actual query gate. So I'll drop this on, duplicate this once more, and we'll have a view to explore here in a second. Now, again, T-SNE is really the bottleneck here in terms of the calculation time. It takes a long time to perform this calculation. FlowSum can be performed on more events, but I want to use both in conjunction with each other during my discovery process, so I can have a dimensionally reduced data space to project and view those FlowSum calculated cluster populations within, but we'll get there. And then I'm going to do one that's going to be FlowSum. Sum is 2. So unfortunately, I'm kind of in a holding pattern until I do that, and I can select these different parameters. Do we have any questions that anyone might type? Okay, then just give me a sec, and we will get there. Actually, while that is calculating, I'll show a few things and then come back to it as soon as it's done. So I mentioned that the FlowJo Exchange is where you get these plugins. So you can link to the plugins at the FlowJo Exchange by clicking on the plugins menu. It's the first list at the top, FlowJo Exchange. That brings you to our GitHub website. And if you want to download and utilize FlowSum, it's pretty easy. You just go down below to where you see available FlowJo plugins and click on the link there. In this case, FlowSum. It downloads a zip file to your hard drive. And then if you click on that zip file, what's included there is the FlowSum jar file. And this FlowSum jar you need to put into the plugins folder for FlowJo. So that is located under applications in this case. And then there's a plugins folder down here and contains all of my plugins. This is where FlowJo actually looks for the plugins and presents them in that list to you. There's also, for any plugin that you download, a how-to document which gives you a little background on the plugin, references as available, and some installation instructions, including the source code of the information, the text that you need to copy and paste into the R environment in order to download and install the packages that run this algorithm within R. So following R, if you need to know how to set up R, I would recommend initially going to your question mark icon up in the top right-hand corner. You can see FlowSum is completed, or sorry, tcne is completed here, and I'll get back to that. But in order to set up the plugins and connect to R, we have documentation available from this question mark link. It'll bring us to our searchable doc site. And if you search for plugins, the very first hit will be how to install these plugins and how to link your FlowJo installation with the R environment, pointing in your preferences to the R installation wherever you've installed it in your computer. And then I have the packaging installation scripts for the released plugins that come with the download, including flow means and spade and cell ontology. So those are the first two steps to get started with FlowSum clustering or other R-based plugins. Go to your question mark, search for plugins, read through the installing plugin site, and I'll give you my email again, timc@flowjo.com, if you have any questions and need some help with the setup. Then you can go to your FlowJo Exchange and get the actual Java files and install using the script in the how-to document that comes down with your download here. So now that that is done, my DC stim has finished calculating, and now I have new parameters. So I can go down here and select X versus Y, and it redistributes into this data space all of the events that were in that concatenated file. So my color map access here is available as a checkbox, and I can now select any of the parameters that I'm looking at. So here is TNF alpha, so it's over here. Where is my CD83 expression? It's all over the place. Where is my CD123 plasmacytoid cells? It's going to be over here, a little bit brighter. And what I'm going to do here is just make a query gate, right? I'm going to go select L1 beta. Yeah, it's over here. So I'm going to look at this area that has high L1 beta expression, and I'm going to make a little gate using my pencil tool and call it query. And that gives me an idea of a frequency there, obviously. But now I'm going to take my stem and no stem gates and drag them onto my query gate and ask, well, in this query gate region, yes, all of the events are coming from the TLR stimulated sample. So whatever this phenotype is, it is enriched in the TLR stem and absent in the no stem very clearly. So to get the multi-parameter phenotype, and here I'm going to select these plots, double click to open up my graph definition or properties window, and select the X and Y axes here so I can see what's going on a little bit better. Sometimes I like to look at the T sin E as a density plot when I'm just looking at the total overlay there, or everything on the same plot, and click apply. This shows me now the space that I'm looking at. I'll get to your question here. I see somebody's asking a question. I'll get there in just a sec. Zoom in here so I can see the difference. Where is the no stem samples occupy this region and the stem samples occupy that region. Obviously we have a query gate here, and if I take that query gate and overlay it onto the rest of the all events, then it creates an overlay. Color coded. If I right click on that overlay down at the bottom, this is where you can make multi-graph overlays. And I'm going to choose the histogram option and then right click once more on those histograms. Oh, my mouse is giving me troubles there. And select multiple parameters. I'm just going to again choose all of my phenotypic parameters, just like I've been doing for dimensionality reduction and any other clustering tools. And I don't care about the viability or the dump gate. So I'm going to display just those. Zoom in a little bit more and expand this out. So what's the actual phenotype of these events? In my query gate, well, that's DR positive, IL-6 positive, TNF. It's essentially finding the same thing that I found in my other example in my workflow slides, because I'm using the same data set. So even though this position of this query gate and where these events fall after the T sin E run that I just did is different than my example in the slides, that's just because the orientation is different. The actual information about the phenotypic information about the populations that parse out into distinct regions or nodes or populations here is consistent, even though I've done this analysis in parallel separately with the same data. So it is a very robust methodology. Actually, the FlowSOM algorithm won the FlowCop contest for reproducibility. So I've got an idea of where what this gate is, and now I can actually explore that by moving the gate around. So I'm bringing this area up, and I can say, well, what's this over here? What's a different population? Let me go ahead and move it down to this region. That node, that island, is DR positive, 83 positive. It's really high for CD123, but negative for most of the cytokines. So it looks like that would be some sort of plasmacytoid dendritic cell or something, right? Is that node actually enriched under the stem? If I look at the TLR stem gates, yes, 92, almost 93% of the events within this query gate here are enriched in the TLR stem. So cool, we can explore that. I'm going to go ahead and start my algorithm to create clusters using FlowSOM, and then I'll take a look at the question that was posed. Just a second. So I'm going to select from my plugins menu my FlowSOM plugin. Here's the user interface with options. I'm going to go ahead and unselect my viability and my dump gate, and I'm going to say instead of 8 metaclusters, I want to force it to make 20 metaclusters. And you can explore some of the options, but it's okay to just run the defaults and see what you get to start. Here's the FlowSOM tree. I'm waiting for it to calculate, so I've got that calculating node. And when it returns populations, I'll be able to drag that in. So here's all the FlowSOM populations that were created, FlowSOM pop 0 through 19. And the tree can be viewed just by dragging and dropping the node that's created after it's done calculating into my layout editor. So here is the minimal, it's the spanning tree relationship between those metaclusters that are in different colors. And then each metacluster has a whole bunch of sub-populations that are displayed in these radial plots with phenotypic information. But my goal here is essentially not, you can relate back to the map, which it can be interesting once you see what's going on, but my goal is really to compare the clusters and ask, do they go up or down? Again, I haven't gated for anything other than cleaning up the data, and now I'm going to find out whether these populations go up or down. So I'm going to copy my no, stim, and stim gates. Copy and paste. And then I'm going to go through each one and I'm going to classify. So here I'm going to make a label that says increased in TLR stim, and another label that's going to be decreased, and a third one that stays the same. So decreased in TLR stim, or let's say increased in no stim. And then no change. Right? So pop 1, is it increased or decreased in the stim? It's increased in the stim. It's got 92%, or pop 0, that is. There we go. Pop 1, increased in the stim. Drop it on, making that overlay. Same thing, I'm just going to continue down the line, make sure I drop them onto the right ones, and then I get to stuff eventually that's going to be increased in the stim population, or in the no stim population. Yeah. 35 to 46. Like, where do you set the threshold? It's really arbitrary, right? I'm going to say this is still up, but it's not as distinctly induced as others. Right. A whole bunch of stuff that's up, not a whole much that's down, because I think the phenotypes get more complex as you stim it. All right, so that's fine. I'm going to go ahead and drop all that on there. Go with a dot plot. I'm going to change these to the X and Y parameters. And then go ahead and right click and say that this is going to be histograms of all of those parameters. Right, so here's the phenotypes that I've just found for a bunch of populations that are up in stimulation. I'm not going to belabor the point because I only have 5 minutes here with you guys still. But these are all the populations that I found, right? They're very complex. Some are high, some are low, some are distinct, like the CD123 population that's very, very much upregulated upon stim. And some of them have very intermediate or, low levels of expression. And my general observation of this type of approach, that you get a bunch of complex phenotypes that may or may not be conducive to manual gating identification, right? So you're actually finding things that have intermediate levels of expression, and if you were manually gating for that particular phenotype, I'd be cutting off through a lot of these populations by setting plus or minus gates for each individual marker, and then doing my traditional Boolean gating or combo gating approach to look at all the possible phenotypic combinations of those markers. So you do get unique or novel populations that you probably wouldn't be able to fully identify using standard gating approaches, using a clustering algorithm approach like FlowSum. And ultimately, I know that these populations are changing, right? That this is what's being induced. I can visually assess and call the phenotypes one by one. But for the numbers to come out, I find it useful to collapse all these populations. So I select everything. And I'm going to use a collapse selection tool, which is under the Edit tab. and then grab all of these populations and export them and bring them back in. So I'm going to go to my file, export the populations. Doesn't matter where I put them. Go ahead and put them in something. And export all those populations. They will be loaded back up into my existing workspace. And so here are now 25 files that I have, and I can make a group and put those exported populations in there. So I'm going to call exported my new group, and I'm going to say if the file name $FIL is the keyword for file name in the FCS standard. So if the file name contains the word export, then go to this group called exported. So create that group. There's my exported files, all 20 of them that have the number of events that were in the FlowSum population. And now I can go ahead and use my statistics tools here under the workspace tab. There's a statistics band. I'm going to add medians of all compensated parameters at the very bottom. All compensated parameters. Oh, maybe I didn't get my compensation matrix. All right, I'm dumb. I'm sorry. I'll do it this way, where I'll grab my compensation matrix and drop it on. Sure. Oh, no. Okay, well, let me save this. I'm just going to open it up again. I think something went wrong when I was dragging and dropping some of those populations. Anyone wants to know about cell ontology that worth trying? Okay, Howie, the answer is no, unless you're very specific about how you name your gating. The answer is that cell ontology, it works, but it only is using specific cell ontology Go classification terminology. And so being able to query that database, it's almost just as easy to go to the database and type in the string of antigens and get your results back using Go from a web browser, as opposed to doing it in here. I've tested it out, and it only works if you're actually using the specific names that are in the Go terminology database for your gating structure. So not always worthwhile. But certainly going to Go and querying what the phenotypes that we find are would be worthwhile, because that's essentially what we're going to do and try and figure out, right? So let's see, I've got my concatenated files, got my exported files. Get my matrix on there. All right, now I'm going to do all compensated parameters. Medians of all compensated parameters. Right, so I get all of my numbers, and I don't care about VIA or my dump. And here I can batch this into the exported group, like a group apply the median gate so everybody has it. Go to my table, create a new table, and drag those statistics in. And then I'm going to highlight all the statistics. And actually, it's always worth exporting the actual frequencies here. So we can grab the gating for positive and negative on our concatenated file down at the bottom somewhere. My no-stim and TLR stim gates, I'm going to drag those to my exported files as well. And so now I've got TLR stim and no-stim gates, which I can enumerate. And I'll go ahead and select everything, visualize and heat map it, and then render that table to display here. And you can see that we end up with all of the populations and all of the phenotypic markers and median fluorescence intensities. And so then you go through and say, well, what goes up and down, classify again, that these are the genes or these are the populations, obviously, that are increased in TLR. These are the things that are increased in the no stem, and those are the population phenotypes for each one. And so in the end, I take that over to Excel and call the phenotypes in a more complex way. So here I've taken my heat mapping in Excel and I've converted it into a plus minus annotation for the populations that I found to either be increased under TLR stem, decreased under TLR stem, right, increased in the no stem, or no significant change. That is, I'm defining it as less than 10% shift between stem and no stem. So that's the workflow. I mean, I hope it's helpful. I think it can be applied to any experimental conditions where you're trying to compare sick versus healthy, stimulated to unstimulated, or even multiple conditions, and trying to understand how do the phenotypes change. And the key difference here is that, sure, I can go through and make a huge gating tree and look for what I know, but I'm inevitably going to miss things that are complex phenotypes with intermediate levels of expression that might actually be valid transitional states, or memory subsets, or even skewed populations that are associated with disease. Obviously disease and response to that disease changes the immune system, and we see that as a change in receptors that define lineages and other populations, as well as upregulation of effector molecules like cytokines that I'm looking at in this context of the dendritic cell populations. But again, when we go back to our workflow and just think about it once more, the only reason I'm dimensionally reducing is to visualize within this space. I'm not actually using this space to cluster. Those are independent algorithms. The flow sum does not depend on the T-SNE, and the T-SNE does not depend on the flow sum. But together they can be used to understand where's the co-expression. It's nice to verify that the flow sum populations that you derive are actually clustering together within a T-SNE data space. And then no matter what you do, you will need to, in order to run this type of approach, you would need to concatenate the populations together that you want to compare. And so that's multiple time points for a single patient, or if that's single time points for a lot of healthy versus a whole bunch of healthy versus a whole bunch of sick people, you're going to have to merge those samples together. And why merge crappy data that's dead debris stuff? So you might as well clean up gate first before you do the concatenation. And bottom line is that by knowing with downsample, you normalize the number of events going in from each sample file and or experimental condition, and that's what that knowledge gives you the power to use to gate on experimental condition and determine whether these populations go up or down by copying and pasting the experimental condition gates onto the cluster populations that you've derived using FlowSum or some other clustering algorithm. And if that's your endpoint, that you're trying to find things that you didn't know you were looking for and determine whether they change between experimental conditions, this type of workflow can certainly aid in that discovery and identification process. So with that, I'll say thank you. My name is Tim. My email here, timc@flojo.com. If you have questions or comments, you're welcome to reach out to me, or